Okay, let's unpack this. Trying to keep tabs on marine animals,
Hmm.
especially elusive ones like beluga whales in their icy remote homes, has always been, well, a real puzzle.
It really has. And the old ways, sending out ships or flying planes overhead, they just have limitations.
Yeah, you can't cover huge areas easily, and let's be honest, a noisy ship isn't exactly uh, blending in. It disturbs them.
Right. But what's fascinating here is that we're now looking at a potential revolution, really, in how we do this. And that's thanks to very high resolution satellite imagery.
Eye in the sky stuff.
Exactly. Just imagine an eye in the sky that can scan vast ocean stretches without disturbing a single creature. The scale of sort of non-intrusive monitoring we could achieve is truly game-changing.
Absolutely. But here's where it gets really interesting. We get these incredible snapshots from space, right?
Hmm.
But turning those pictures into reliable counts, like actual population numbers,
Mhm.
it isn't as simple as just looking.
No, not at all. The standard method for automated detection uses deep learning, which is powerful, but it usually demands this massive amount of manually created annotations.
Right? Annotations, like someone has to sit there and draw precise digital boxes around every single whale.
Yeah.
Thousands of images.
That's exactly it. You can imagine how painstaking, how time-consuming that is.
Talk about a bottleneck.
Precisely. So the big question becomes, how do we expand our monitoring capabilities to the scale we actually need without getting completely bogged down in this annotation problem?
And it's not just the sheer number, is it? You mentioned something about fuzzy images.
Yeah, that's another layer. Think about those tricky images. Maybe a whale is just below the surface, or the water's choppy, or there's glare.
Hmm.
Researchers call these uncertain whales, and they often just get excluded from studies because they're hard to label confidently.
So you're missing data. It's like
Wow.
trying to count sheep in fog. You're bound to miss some.
You are, and that could give us an incomplete picture of the population.
Right. So our mission in this deep dive is basically to explore a new automated system that tries to solve these exact problems.
That's the plan. We're going to look at how it aims to detect not only those crystal clear beluga images, but also the more ambiguous ones, and harp seals too, all using satellite data.
Okay, and our source for this is a really compelling research paper detailing this innovative approach.
Yep, let's dive in.
So this annotation bottleneck, the traditional way sounds like a nightmare.
It can be. You need highly skilled people spending countless hours drawing these really precise boxes around each animal in image after image. It's slow, it's expensive, and honestly, there can be inconsistencies between different annotators.
So this new study, they tackled this head on.
They did.
Okay, so what's their clever workaround? How do they escape the endless box drawing?
They came up with a really ingenious two-part solution. First, instead of drawing those full bounding boxes, they mainly use point annotations.
Point annotations, like just clicking.
Essentially, yeah. Imagine just clicking on the center of each whale in the image. That's basically it. It's dramatically faster and much less work than outlining the whole animal.
Okay, that already sounds way better. What's the second part?
The second part is where the AI magic comes in. They bring in this incredibly powerful model called the Segment Anything Model, or SAM.
Sam. Okay, tell me about Sam. What does it do?
So SAM is a very advanced AI. It's what's called a foundation model, trained on this enormous data set, like billions of images and masks. Its superpower is that it can identify and segment, meaning outline, virtually any object you give it a prompt for.
A prompt, like asking it to find something.
Yeah, and the prompts can be super simple, a point like we just talked about, or maybe a rough box or even text. Because it's seen so much data, it's remarkably good at figuring out the boundaries of things it might not have been specifically trained on before, like, a beluga whale seen from space.
Wow. So instead of humans drawing hundreds of boxes, you just click.
Mm-hmm.
And Sam does the hard work of outlining.
That's the core idea. It massively reduces the human effort involved.
Okay, so they're clicking on the whales, providing these point annotations.
Yeah.
How does Sam take those simple clicks and create the detailed outlines needed to train the actual detection system?
Good question. So the researchers feed Sam that point annotation, basically saying, hey, Sam, there's something important right here.
A little pointer.
Exactly. And to give Sam a little extra context, especially about the rough size of the animal, they also added a small buffer zone around each point. For belugas, it was a 4-meter square, and for the smaller harp seals, a 2-meter square.
Okay, like giving Sam a little hint. It's around here, and it's roughly this big.
Precisely, just giving it a nudge, focusing its attention in the right area.
Makes sense. But what happens when you have a bunch of whales all close together? Like a pod? Wouldn't Sam end up drawing masks that overlap all over the place?
That's a critical point, yeah. And they developed a smart way to handle it. When the initial masks that SAM generates do overlap in these dense scenes, they use something called an overlapping pixel assignment algorithm.
Okay, sounds technical, but what does it actually do?
Basically, for any pixel that falls into the overlap zone between two or more masks, the algorithm checks how close that pixel is to the original point annotation for each nearby whale.
The click point.
Right. And the pixel gets assigned to the mask belonging to the whale whose point annotation is closest. It's a clever way to detangle the overlaps.
That is clever. So it ensures each pixel belongs to just one whale, even in a crowd.
Exactly. It creates these nice, clean, non-overlapping individual masks. It's a key step for getting accurate counts in dense groups.
Okay, so Sam draws the shapes based on a click and a hint. This algorithm sorts out overlaps.
Yeah.
What's the next step? Are those masks the final product?
Almost. Once they have these precise pixel-level masks from SAM, they convert them into standard bounding rectangles. These rectangles then become the training data for their main object detection model.
Got it. The rectangles are what the detection model learns from.
Yep. And what's really interesting and important for checking the quality is that the researchers also had human experts manually refine a portion of these SAM-generated boxes.
So they could compare the purely automated ones with ones that had a human double check.
Exactly. It was essential for evaluating just how accurate the automated annotations actually were.
Okay, so Sam helps create the training labels much faster. Now, how do they actually find the whales in new satellite images? What's the detection model they used?
They chose to use YOLO, which stands for You Only Look Once.
Catchy name.
It is. And it's a really promising type of object detection algorithm. It's been pretty successful in spotting various things in images, including marine animals and other studies. Specifically, they used a version called YOLOV8s.
YOLOV8s. Why that specific one?
It's known for striking a good balance between speed, how quickly it can process an image, and accuracy, how well it detects the objects. You often have a trade-off there, and YOLOV8s is kind of in the sweet spot.
Okay, YOLO. Yeah. You only look once. How does it actually work its magic? Said it's fast.
Yeah, the name gives a hint. Unlike older methods that might look at an image multiple times or in multiple stages, YOLO looks at the entire image in a single pass.
One look.
Right. It divides the image into a grid, conceptually. And for each part of that grid, it simultaneously predicts a few things. Potential bounding boxes, a confidence score for each box, like how sure it is there's an object there, and the category of the object is a certain whale, an uncertain whale, or a harp seal, all at once.
Wow, okay. That sounds efficient. So what data did they feed this YOLOV8 model to teach it what belugas and seals look like from space?
They used a collection of satellite images, specifically cropped sections, taken over known beluga whale summer habitats in northern Canada.
Where exactly?
Clearwater Fjord and the Churchill River area. The images were from 2021 and 2022.
And the satellites. You mentioned high resolution earlier.
Yeah, very high resolution. They used WorldView 3 and WorldView 2 satellites. These give incredible detail, down to 0.3 meters and 0.46 meters resolution, respectively. You can really see individual animals.
That's amazing detail. How many animals did they start with in their data set?
Their initial manual point annotations identified a good number. 577 certain whales, 407 of those trickier uncertain whales, and 542 harp seals.
Okay, a decent amount to learn from.
Definitely. And having this faster SAM-based annotation pipeline means researchers could potentially process way more data in the future, covering larger areas or longer time periods than was feasible before.
Right, scaling up is key. So they had these images. Do they need to prep them further before feeding them to YOLO?
Yes, a few standard pre-processing steps. First, they converted the images to an 8-bit format, which is common for image processing. Then, because the original satellite images are huge, they broke them down into smaller, more manageable patches.
Smaller chunks.
Yep. Either 320 by 320 pixels or 192 by 192 pixels, depending on the original image resolution. And crucially, they only kept the patches that actually contain the animals they were interested in, the whales or seals.
Filtering out empty ocean.
Exactly. That resulted in 538 relevant image patches for training and testing.
And how do they ensure the model learned properly and, didn't just memorize the specific images they showed it?
Good point. They used a standard practice of splitting their data set. 70% was used for the actual training, 10% for validation that helps tune the model during training, and the remaining 20% was held back as a final test set.
The test set being images the model had never seen before.
Precisely. That gives you a true measure of how well it generalizes. They also use data augmentation.
What's that involved?
It means creating slightly modified versions of the training images, things like flipping them horizontally, adjusting brightness or contrast slightly. It helps make the model more robust, less sensitive to small variations, and prevents overfitting.
Makes sense. Anything else?
Yes, they also use transfer learning.
Ah, I've heard of that. It's like giving the AI a head start.
Exactly. Instead of starting the YOLOV model from scratch, knowing nothing about images, they initialized it with weights that were pre-trained on a massive general-purpose image dataset called MS-CHEM-O. It contains millions of images of everyday objects.
So the model already has some basic understanding of shapes, textures, edges.
Right. It has a foundational visual understanding, which it can then adapt more quickly to the specific task of recognizing whales and seals. It usually leads to better performance and faster training.
Okay, so they had these three different ways of creating the training labels, right? The buffer zones, the manually checked sandboxes, and the raw sandboxes. Did they train YOLOs separately for each?
Yes, exactly. They compared 3 different training scenarios to see which annotation method worked best.
Can you remind us what they were called?
Sure. First was YOLO Buffer. That model was trained using those simple fixed-size square boxes created around the initial point annotations. Kind of a baseline, simpler approach. Second was YOLO Box. This one used the bounding boxes that were initially generated by Sam, but then manually checked and refined by the human experts. Think of this as the gold standard comparison, high-quality labels, but with that manual effort involved.
Right, the benchmark.
And finally, the key one, YOLO SAM. This model was trained using the raw, untouched bounding boxes generated directly by Sam using just the point annotations and buffer hints. This is the real test of the fully automated annotation pipeline.
Got it. So YOLO Buffer, YOLO Box, and YOLO SAM. Ready to see how they did.
All right, let's get to the exciting part, the results.
Yeah, how did these different training approaches actually compare in finding the whales and seals?
Well, first, it's interesting to look at how much correction the SAM-generated labels needed from the experts for that YOLO box model.
Right. How good was SAM on its own?
Pretty good, actually. The experts only needed to manually correct about 19% of the SAM labels for the clearly visible certain whales.
Okay, so SAM got it right over 80% of the time for the clear ones.
Yep. For the more ambiguous, uncertain whales, the correction rate was a bit higher, around 21%.
Makes sense. They're harder to see.
Exactly. And interestingly, for the harp seals, only about 4% of the labels needed manual correction.
Wow, so SAM found seals really easy to outline accurately.
It seems so. Likely reflects maybe their shape, contrast, or how they group together in the imagery compared to the whales.
Okay, so SAM does a decent job, especially on seals and clear whales. Now, how did the actual detection models perform? The YOLO models trained on these different labels?
They primarily measured performance using the F1 score.
F1 score. Remind us what that balances.
It's a standard metric in detection tasks. It balances recall, what fraction of the actual animals did the model find, and precision of the animals the model said it found, how many were actually correct. A higher F1 score means better overall performance.
Got it. So the result.
The YOLO SAM model, the one trained on the fully automated SAM labels, achieved a really impressive overall F1 score of 72.2% for all whales combined on the test data.
Over 72%. That sounds pretty solid for an automated system.
It is. And for harp seals, its F1 score was 70.3%.
Also strong. So how did YOLO SAM, the automated one, stack up against YOLO Buffer, the one using those simple square buffers?
This is where the benefit of SAM really shows. YOLO SAM significantly outperformed YOLO Buffer in terms of recall, especially for the certain whales and the harp seals. It found more of the animals.
Why is that, do they think?
The researchers believe it's because the SAM-generated boxes are much more precise. They follow the actual shape of the animal much better than a fixed-size square buffer.
Ah, so the buffer might include too much empty water or cut off part of the animal.
Exactly. Especially in crowded scenes, those square buffers can overlap badly or merge animals, while SAM's tighter masks help distinguish individuals better, leading to higher recall. YOLO SAM was also more accurate overall across all categories compared to YOLO Buffer.
Okay, so SAM's precision pays off in detection. Now, the big comparison. How did the fully automated YOLO SAM do compared to YOLO Box, the one with the human-refined labels?
This is another key finding. The average F1 score of YOLO SAM was remarkably close to YOLO Box.
Really? So the fully automated pipeline performed almost as well as the one with manual expert checking?
Pretty much, yeah. It strongly suggests that this automated pipeline, just using simple point annotations and letting Sam do the heavy lifting for labeling, is a genuinely viable alternative to spending all that time on full manual annotation.
That's a huge takeaway. It gets you extremely close to human-level annotation quality, but way, way faster.
That's the implication. It dramatically lowers the barrier to creating large, high-quality training data sets.
But you mentioned earlier the uncertain whales were trickier for SAM to label. Did that affect Yellow SAM's detection performance for them?
It did, slightly. Yellow Sam had a lower F1 score specifically for those uncertain whales compared to the other two models, YOLO Buffer and YOLO Box.
Why was that?
It likely stems directly from the lower quality of the initial SAM annotations for those ambiguous targets. Remember, SAM itself struggled more with them, leading to that higher manual correction rate.
Yeah.
So when those uncertain whale labels were manually refined for the YOLO Box model, the detection performance for those specific whales did improve, getting closer to the level of the YOLO Buffer model.
Interesting. So for the really tough, fuzzy cases, a bit of human oversight on the labels still helps the final detection.
It seems so, yes. But for the majority of cases, the automated SAM labels were sufficient to get near expert performance.
Got it. What about the differences between detecting whales and seals overall?
The study consistently found that detecting harp seals was generally easier, higher F1 scores than detecting both the certain and the uncertain whales.
Any guesses why?
Could be a few reasons. Maybe the seal's smaller size, their tendency to haul out on ice floes in larger groups, making them distinct, or perhaps just better contrast against the background and the specific images they had. Whales, being larger, sometimes partially submerged or in variable water conditions, might just pose a slightly tougher detection challenge.
You also mentioned the shape of the bounding boxes being important. Can you elaborate on how YOLO SAM's boxes looked compared to YOLO buffers?
Yeah, this is visually quite striking in the paper. The bounding boxes produced by YOLO SAM, because they were derived from SAM's segmentation masks, very closely matched the actual outline of the whale's body like a snug rectangle around the segmented shape.
Okay, tight fit.
Whereas YOLO Buffer, using those fixed square boxes centered on the point annotation, often included a lot of surrounding water or ice that wasn't actually part of the animal.
Just empty space inside the box?
Right. And that extra background noise within the training box can potentially confuse the model, making it harder for it to learn the key features of the animal itself.
And you said this helped in dense groups too.
Yes, because YOLO SAM's boxes were more precise and less likely to overlap unnecessarily, it resulted in fewer misdetections, better recall in those tightly packed groups where the fixed buffer boxes might just merge into one big blob.
Makes total sense. Okay, lastly on results, did the paper mention the kinds of mistakes YOLO SAM did make? Where did it get confused?
Yeah, they looked at the confusion matrix. There were some instances where the model confused whales or seals with the background, and vice versa, missing animals or hallucinating them.
What might cause that?
The researchers suspect environmental factors in the satellite images played a role. Things like waves that might momentarily resemble an animal's shape, sun glare on the water, maybe even rocks near the shore that could look a bit like a seal from orbit.
Tricky conditions.
And even things like mild cloud cover could sometimes obscure the animals just enough to cause the model to miss them. It highlights that even with advanced AI, the quality and clarity of the input imagery is still crucial.
Okay, so we've seen how this new system works, the really impressive results it achieves. Now for the big question, so what? Why is all of this significant? What are the broader implications?
Well, the implications here are truly exciting, I think. The immediate advantage is clear. Using SAM-labeled bounding boxes gives you training data that better captures the animal's shape, cuts down on background noise in the box, and handles crowded scenes much better.
Leading to better detection.
Leading to improved recall, primarily. We're less likely to miss animals, which is critical for accurate population counts. And maybe just as importantly, this automated approach dramatically cuts down the manual effort needed for annotation.
Freeing up researchers' time.
Hugely. It frees up valuable time and resources. This speed boost isn't just a small convenience. It could genuinely allow researchers to analyze far more satellite data than ever before. Think about covering much larger geographic areas or monitoring changes over longer timeframes. The scale difference is potentially massive.
That really is huge for monitoring these vast remote ocean habitats more frequently and effectively. What other possibilities does this open up beyond just counting belugas?
The potential reach is quite broad. This core technique, using a simple point click with SAM to generate detailed labels, could likely be adapted to detect other marine species.
Like what?
Well, things like narwhals, maybe even polar bears if they're visible on ice floes in the satellite images. And it's not just limited to satellites. The same approach could probably be applied to high-resolution aerial photos from drones or planes.
Expanding the toolkit for wildlife monitoring.
Definitely.
And it's not just about counting animals, is it? You mentioned something about shape.
Exactly. This is another really fascinating potential application because SAM generates these detailed pixel-level masks, the actual outlines of the whales.
Textures, rectangles.
Right, the actual shape. It opens up the possibility of automatically estimating their biometrics.
Biometrics, like measurements.
Yeah, things like their body width and lengths derived directly from the segmented mask in the satellite image.
Wow, from space. You could measure a whale.
Potentially, yes. Getting that kind of morphological data automatically across large populations would be incredibly valuable for ecological studies. You could track growth rates, assess body condition, understand population health trends, things that are very difficult and expensive to measure using traditional methods.
That's incredible. So a simple click leads to not just detection, but potential measurement too. That feels like a real leap.
It really does. It connects the large-scale observation power of satellites with detailed biological information.
So if someone listening is really intrigued by this, maybe a researcher or just a tech enthusiast, and wants to delve deeper or even try this out, is that possible?
It absolutely is, and this is great. The researchers have generously made the code for their entire pipeline, both the SAM-based labeling part and the YOLO detection part, publicly available.
Oh, fantastic. Open source.
Yep, it's on GitHub. The link is github.com/VoyagerX, VoyagerX Beluga Seeker. They're actively encouraging the broader research community to explain it, use it, and build upon their work.
We'll make sure that link is available. That's excellent for transparency and collaboration.
It really is.
Okay, so to bring it all together then, what's the main takeaway from this deep dive into using point labels and SAM for tracking marine mammals?
I think the core message is that this study represents a really significant step forward in automating marine mammal monitoring. By cleverly combining simple point annotations, which are fast to create, with the remarkable power of the segment anything model, they've developed a highly effective way to generate high-quality training data.
Training data for what?
For detecting beluga whales and harp seals in challenging satellite imagery. And the key benefits are, 1, it massively reduces that laborious manual annotation effort. 2, it boosts detection accuracy, especially in tricky situations like dense groups or for those harder to see uncertain whales.
And the potential beyond that?
And 3, it holds tremendous promise for wider applications, other species, other types of imagery like aerial photos, and even estimating those valuable biometrics for ecological research and conservation.
Truly groundbreaking stuff. It really feels like we're on the cusp of a new era in how we observe and understand these incredible, often hidden creatures.
I agree. It's a very exciting time at the intersection of AI and remote sensing for ecology.
And on that note, here's a final thought for you, the listener, to maybe mull over. Given these rapid advancements in both the availability and the sheer resolution of satellite imagery, how might these automated detection and biometric estimation techniques fundamentally reshape our understanding of marine animal populations? How might they change how we track the impacts of environmental change, like melting sea ice, on them? And to maybe even think about what other unexpected applications might pop up from this powerful combination of AI looking down from space, all in our ongoing efforts to better understand and protect the planet's wildlife. Definitely something worth pondering.